back again. Glad you're here. We're going to continue talking about the nervous system. I know you're surprised to hear that. Waiting for my... Continuing to talk about the nervous system, specifically digging into what's happening at the synapse. Make sure that you guys are seeing what I'm seeing there. Yes. Okay. The synapse. Kind of the big deal, the big thing, the stuff of the synapse is neurotransmitters. Okay. As we've already discussed a lot, right? So if we look at our neuron, we have our cell body with our dendrites. Okay, hopefully looking really, really, really familiar at this point. We're just going to go over kind of what we've touched on so far, what we've talked about so far, and review that before we move on. So here at the cell body, we have a graded potential. Get a graded potential at the cell body. And then from a graded potential, two different things can happen, right? We can have is threshold met? No. Or yes. Right? At the axon hillock, the question is, are we meeting threshold? So if we meet threshold, then what happens? What happens is an action potential. That domino falling reaction. If we don't, let's see, if we don't meet threshold, nothing happens. The neuron doesn't do anything. Okay, so that's not really an interesting path to follow. Okay, if we get an action potential, then that's carried down the axon to the axon terminus. And what does that do? If this neuron is interacting with another neuron, those are my dendrites. If this neuron is interacting with another neuron, then this action potential results in the release neurotransmitters. I'm trying not to use the same colors over and over again. Then this action potential results in the release of neurotransmitters, right? We're releasing neurotransmitters. Okay. That release of neurotransmitters then leads to what on this other neuron? If the target cell is another neuron, then what do we get over here? We get another grade. Oh, I could use the same color. That would be useful, wouldn't it? Boom, we get another graded potential. Okay. And so our focus for this discussion is going to be right here. synaptic transmission, and then the resulting graded potential. So just to kind of review where we've been, give you a little preview of where we're going. Oh, and hey, there's some water. Okay, so we're going to go all the way down here, and we're going to be focusing on the axon terminal. So I'm just going to draw one sort of bud, you want to call it that, just to simplify things. And so this is the axon terminus. This is where we are at the axon terminus. And so, or the axon terminal. And so what happens to get synaptic transmission? The first thing that has to happen, the thing that we've been focusing on this whole time, is an action potential, right? So the action potential comes down the axon. So we have the action potential. Okay, first thing that has to happen. What this does, this action potential, what it does is, yet again, we have some voltage gated channels, but now we're going to introduce some different voltage gated channels. These are new. We haven't talked about these guys yet. So I don't think we've talked about these guys yet. Okay, so here in the axon terminal or axon terminus, we have a new voltage gated channel, and this is a voltage gated calcium channel. Okay. Depolarization, yet again, we have a voltage gated channel. This is a voltage gated calcium channel. The depolarization causes that calcium channel to open. In this case, we have quite a lot more calcium outside of the cell than we do inside of the cell. So if we start with more calcium outside of the cell than we have inside of the cell, where's that calcium going to go? That calcium is going to go down its concentration gradient into the cell. Okay, so we have calcium entering the cell at the axon terminal or axon terminus due to the depolarization and the opening of voltage gated calcium channels. Okay, so now we have a bunch of calcium in our cell, calcium now flooding into the cell. And what calcium does is that it triggers what's exocytosis, you may remember this from earlier lectures, of synaptic vesicle contents. So what the heck does that even mean? It means that hanging out here in the axon terminus are these vesicles. Okay, so vesicles, it's like a bubble of stuff. Okay, these synaptic vesicles are hanging out in the axon terminus, and they are chock full of neurotransmitter. So they're like kind of like little holding tanks, neurotransmitter holding tanks. And in order to get those synaptic vesicles to come and open their neurotransmitters, open their contents to the outside environment, you have to get an influx of calcium. So calcium comes and what it does is it triggers these vesicles to come and sort of like their stuff out into the external environment. Those vesicles bond to a docking protein in the membrane, in case that interests you. So the depolarization facilitates the influx of calcium. The influx of calcium is necessary for the synaptic vesicles to release neurotransmitters into the synaptic cleft, into that space. So remember, we have a space between this axon and the next one. If we're talking about an axon, it doesn't have to be an axon. For right now, we're gonna say it is. Okay, so let's imagine this. Here's our dendrite of our other axon. Okay, so those neurotransmitters get dumped into that space. Oops. And it's going to diffuse across this space. So it's going to diffuse across this space. And once again, this dendrite of this next neuron has to have what? What does it have to have? Something really, really, really critically important. It's got to have the receptor, right? Okay. We'll talk more about that later too, different receptors for different types of neurotransmitters. We'll talk about that briefly in this lecture, but then we'll dig into it more later. Okay, so the neurotransmitter diffuses across the synaptic cleft. It's going to bind to the receptor on the dendrite of the next neuron. In this case, the neuron is the target cell. It does not have to be a neuron. So what this neurotransmitter binding does is it initiates a response, right? It initiates a response in this cell. And so these next steps are kind of everything that we started with. So we started with our graded potential, went through our action potential, now we're at synaptic transmission, and now we're back to what? A graded potential. We have a cellular response. If this is a neuron, if it's not a neuron, there's a whole other host of cellular responses that can happen that some of which we've talked about previously. Okay, so neurotransmitter binding initiates some sort of cellular response. In the case of a neuron, what we've talked about so far is a graded potential, right? So we can get a graded potential. In the case of a neuron talking to another neuron. Okay. The cool thing about graded potentials that we haven't touched on yet is that they don't always excite the neuron. They can actually inhibit the neuron. What do I mean by that? So far, when we've talked about graded potentials, our goal was to get to an action potential, to talk about what an action potential is. So if we wanted to get an action potential, then the response of this secondary postsynaptic neuron, let me explain what that means. So this is a presynaptic neuron. And this is a postsynaptic neuron. Because one of them is before the synapse and one of them is after the synapse. Okay, so this postsynaptic neuron, if we're trying to get an action potential, this neurotransmitter binds to a receptor for trying to get an action potential to happen. What do we want that graded potential to do to the interior of the postsynaptic neuron? What do we want to happen? So let's think about the environment, the resting membrane potential, if you will, of this neuron. Our resting membrane potential, I can't remember, let's use blue. I keep changing colors on you guys. Y'all really, really can't trust my colors, can you? Okay, so remember that the interior environment is negative relative to our outside environment. And in order to trigger an action potential, we have to get that internal environment to negative 55 at the axon hillock. So if we're trying to trigger an action potential, if we're trying to get that neuron to fire, what we want this graded potential to be is called excitatory. So a graded potential, if we want a neuron to fire, that's going to be an excitatory. Is it A or I? I don't know. Excitatory increases the likelihood that a neuron will fire. I'm going to say that an action potential will propagate because that is more sciencey. Incorrect. Okay, an excitatory graded potential makes it more likely that we're going to get an action potential. So if we want to make it more likely that we're going to get an action potential, then whatever this neurotransmitter is doing, we want it to open channels that let positives in, right? So if we want an excitatory graded potential, then this neurotransmitter, the effect that it's going to have on the cell is to open those sodium channels, okay? So an excitatory graded potential, so what it's going to do is it's going to make the interior of the cell more positive, okay? So we go back up here to our drawing. That means that this neurotransmitter is going to open sodium channels. So we're going to get the influx of positively charged sodium. Okay, now we have that graded potential extending through the cell body. And if that's strong enough and we reach negative 55 at the axon hillock, boom, we get an action potential. So what that does is it excites the neuron, okay? It makes it more likely to produce an action potential. So that's an excitatory graded potential. That's pretty much all we've talked about to this point. But the cool thing about graded potentials is they don't only have to be excitatory. Graded potentials can also be inhibitory. Okay, and so if we think about what an excitatory graded potential does, what do you think an inhibitory graded potential? What will cause an inhibitory graded potential? What happens? Okay, an excitatory graded potential, we're trying to make the inside of the cell more positive. So inhibitory graded potential, okay, it decreases the likelihood of an action potential. The way it does that is it makes the internal environment of the postsynaptic cell more negative. Okay, so I'm going to draw this again so it doesn't get super messy. Okay, so here's our dendrite. Here's our presynaptic neuron. It's releasing our neurotransmitter. Neurotransmitter gets released. We have our receptor. Okay, and if this is going to trigger an inhibitory graded potential, what happens when that neurotransmitter binds to that receptor? It's not going to open sodium channels, which has really been our focus. To get to an action potential, we need to open those sodium channels. It's not going to open sodium channels. It can do a couple of things. So instead of opening sodium channels, what it can do is, hello, new channel, brand new thing that we haven't talked about yet. What that can trigger is the opening of, instead of a sodium channel, chloride channels. How do you make the interior of the cell more negative? Add more negatives. So if we get chloride channels open, chloride's going to move down its concentration gradient, and rather than going from -70 to -55, which is what we wanted to do here in an excitatory potential, we want to go, right, here's our -70. We're trying to get closer to threshold. So we're trying to go more positive. Instead of doing that, for inhibitory, here's our -70. Ah, that was terrible, guys. Hang on, I'm gonna fix it. Okay. Here's our -70. So what we're gonna do instead is we're gonna go more negative. So -80, -90, -100, it's gonna become more negative inside the postsynaptic cell. Okay, so if we go more negative, if we go even more negative from -70, think about how much harder it is to get up to -55. So if we go down in that graph here, if we go down here, how much farther do we have to go to get there? So neurotransmitters that cause the cell to become more negative, are inhibitory. They decrease the likelihood of an action potential. Okay, so these are inhibitory neurotransmitters, and both exist. So we have stimulatory neurotransmitters. We have neurotransmitters that are, excitatory is the word that I should have used. I should be consistent. So we have excitatory neurotransmitters, and then we also have inhibitory neurotransmitters. And if you think about it, this makes sense, right? We don't want to only be exciting our neurons. We also want to kind of chill them out sometimes, right? Okay. Oh, so we can let chloride in. Another thing that we can do is that we can let potassium out. Okay, so we let the positive ion out. That's also going to make the interior of the cell more negative relative to the outside of the cell. So either we pump a negative ion in or we pump a positive ion out. Makes the interior of the cell more negative. We make the interior of the cell more negative. It's harder to produce an action potential. Where are we on time? Oh, 23 minutes. Okay, we'll stop there and then we'll come back and we'll actually talk about these neurotransmitters with names and everything.